We can't hear you, Ma.
Good afternoon. Can you hear me?
Yes,
Yes, Ma,
ma'am.
we can hear you, Ma.
Yes, ma'am.
Okay, thank you very much. I am Kumbi Kurumi from Botany Department. Oh, I will be taking you on algae. I'll be treating the topic algae with you. Please, can you see my screen? Please, can you see my screen?
No, ma.
No, ma. No, ma.
Screen is here to show. I think it's loading.
I am sharing screen. No, I have shared screen. But you have my notes, right? I sent it earlier.
Yes, ma.
Please, can you open to the first lecture, algae lecture 1. Are you there?
Yes, ma.
Just a second. So we are going to be treating algae, which is not completely new to you, because we have done part of this in 100 level when you were taking the course BTN 121. Okay. So I also sent the PowerPoint, which is supposed to guide you. So for this particular BTN 211 algae topic, we are going to be going a little deeper. It's nothing difficult.
Ma, we can't hear you again. No.
Please, can you hear me?
Yes, Ma, we can now hear you.
Can you hear me now?
Yes, ma'am.
Okay, so sorry. So the first lecture note is you have the you have the title algae. Algae are chlorophyllous talophytes, that is, they are plants lacking roots, systems, and leaves. They manufacture their own food due to the presence of chlorophyll, which enables them to photosynthesize. Hence, they are autotrophic. Algae do not produce flowers. They range in size from unicellular microscopic forms to multicellular giant kelps, which attain lengths of over a meter. They may also exist as filaments or in colony. So this is not new to us. Please, if you have any issues, please kindly type with the broadcast, kindly type in the chat section, please. In case you stop hearing me or I'm no longer audible, just type in the chat section. I hope you understand that, please. So that I will not just be talking to myself. If there is any issue with the broadcast, please kindly state in the chat session. Thank you so much. Okay, we are not new to algae. Everything I stated a few minutes ago is something we know. They are the first set of plants that produce chlorophyll, and they are called talophytes because they lack true roots, they lack true stems, they lack true leaves. When you come to the lab, by January, by God's grace, you'll be able to see some examples of algae in the lab under the microscope. You will see they are a group of lower plants in terms of the fact that they do not have true roots, true stems and leaves like we have in the higher plants you see around. When you see algae, you are not able to say this is the root, this is the stem, this is the leaf. You can't differentiate them. The parts, you can't differentiate the parts rather from one another. They manufacture their own, and that's why they are called talophytes. The whole body is a thallus. This is just like a reminder of what we did in BTN 121. The whole body of a algae is called a thallus. The whole plant body is a thallus. They are not differentiated into different parts. They manufacture their own food. Fungi do not do that. Bacteria do not do that. The viruses do not do that. Algae are the first group of plants to manufacture their food. So they are autotrophs and they do not produce flowers. If you see your higher plants, you will see that they produce flowers. There is no algae anywhere in the world that produce flowers. Don't come and say, so people don't come to the lab now, they will draw algae, they will not label flower. Algae don't have flowers. They don't produce flowers like higher plants. Now they can be very, very tiny, microscopic forms, and they can be very large, giant scales. Like I said, I have sent you the PowerPoint. You will see images or pictures of examples of unicellular microalgae and the multicellular giant kelps. You will see examples on the slide. When you come to the lab, you will see the microscopic forms. We might not be able to give you the multicellular giant kelps. Now, when I said they can be multicellular giant kelps, that does not mean that they produce leaves. That does not mean that you can see the leaves differentiated from the stem. No. No matter how big or how large rather an alga is, it is still a thallus. The plant body is never differentiated. The plant body is never differentiated. Please, if you cannot hear me, kindly indicate, please, so that I will not be talking to myself. There might be network issues. Please state in the chat session if you stop hearing me at any point, please. So, regardless of how large the alga is, it is still a plant body. It is still a thallus rather. The body is never differentiated into roots, stems, and leaves. They don't have that. The body is still a thallus. So don't say giant kelps, that means it will have roots, stem. None of them, none of the algae has differentiated root, stems, or leaves. Please bear that in mind. Aha. It's very important because some students still make that mistake. They say giant, multicellular. They can be over a meter long. Regardless of that, they are still talophyte. They can be filaments, filamentous, or colonial. So I move on to the range of forms. Unicellular algae is the first group, the colonial algae, the filamentous, the multicellular. These are the different forms in which algae exist. These are the different forms in which algae exist. The first group are the unicellular algae. They exist as single cells. They exist as single cells, just one alga. You will never see them together. This is still trying to remind you of BTN 121. Examples include Chlamydomonas, Euglena, Chlorella. They are unicells, they are single cells. That is how you see them always. You don't see them attached to another cell existing like that. No, they are always single cells. They are unicellular algae. So in the exam, you can be asked, oh, which of the following is an example of the unicellular alga? You should pick your choice correctly, please. Colonial algae include algae that exist in groups. Now, these ones, you don't see them as single cells. They exist in groups, maybe groups of 2, 4, 6, or thousands, just in a colony. Example are your volvox. This one, we have been hearing it in secondary school, volvox. Bandorina, Eudorina, they are colonial algae. You see them existing. The cells come together to form one alga. Single cells come together, they stay together. That is how you always see them. You will never see them as unicells. Do you understand that? Please, can you state if you understand what I have just said?
You understand,
Yes,
ma?
ma'am.
Yes, ma.
Please, can you state in the chat section if you understand what I just said?
Yes, ma.
Thank you very much for your response. I will prefer the chat so that we don't interrupt one another. Thank you very much for your response. Thank you, thank you, thank you. Please, we should take note of this. Exam is not difficult. If you listen, you pay attention in class. But some students still carry over between 211 and you'll be wondering why. Please, let's pay attention. Thank you very much for your responses. Okay, so we have filamentous algae. They consist of colonies of microscopic algae that join together to form thread-like filaments. These ones, you will see them like threads, like chains. You will see them like chains, like threads. They are filamentous. A very good example is your Spirogyra, this one too, since secondary school biology. Nostoc, Oscillatoria, they are all examples. I'm giving common examples, the ones you can easily remember. This is just 200 levels, so we don't want to stress anybody. So at least you will remember Spirogyra, you will remember Nostoc from your nutrient cycle, nitrogen cycle in biology. Oscillatoria shouldn't be new to you. You must have come across it in your 100 level BTN 121. So those are filamentous algae. Now you don't see unicellular algae as filamentous algae. That's why we put them in their groups, in the forms in which they exist. We put them, the way we see them physically, their morphology under the microscope, the form, whether it is a unicellular organism or a colonial organism or a filamentous organism. In terms of algae now, we put them in these groups. Now we have the multicellular algae that are large macroscopic algae. Again, they are still thallophyte. Don't be deceived with the word large macroscopic. This is just telling you that these ones, you don't need microscope to identify them. The previous ones I mentioned earlier, the unicellular, the colonial, the filamentous, those ones you have to put them under the microscope to actually identify them. You can't be looking at chlorella, for example, without microscope and know that it is chlorella. No, it's not possible. You have to prepare a slide, then you will be able to see the morphological structures that actually identifies the organism as chlorella. But for the multicellular algae, you have them in large microscopic form. You are able to see them and identify them without the aid of a microscope. Again, that doesn't mean you're going to see stem, leaves, flowers, roots. They are still thallophyte. Please, I'm saying that again, so that students will not go and lose marks. Not go and lose marks unnecessarily in the exam. Please, which they develop specialized tissues, but lack true stems, leaves, or roots that are found in higher plants. Please, this group of algae may grow up to 80 meters or more. Can you see that? They can be that large, but regardless of how large they are thallophyte. They are usually called seaweeds. You see example of that on my slide. The PowerPoint I sent, you can check that later. I move on to habitat now. Where do we usually see algae? Where do they inhabit? Where are they found? They inhabit a wide range of habitats. They are found in fresh water. Fresh water is not water that is fresh. Don't go and say fresh water is water that is fresh. Fresh water in terms of salinity. Water bodies where the salinity is low, like your rivers. If anybody has been to the river before, the Lagos Highland people may not have an idea. But for people that have lived in villages like me, if you have been to rivers before, if you taste the water from the river, it doesn't taste any salt. You can't taste salt. That salinity is very low. You don't taste salt. Unlike if you have been to the bar beach, if water from the bar beach splashes on your body or your lips, you will taste salt. So your bar beach, for example, is like a marine environment where the salinity is very high. And the brackish environment are maybe like the Lagos Lagoon, for example. The salinity is not too low like you have in rivers and is not too high as you have in marine. So brackish is like in between. So we have different algae inhabiting this aquatic habitat. The freshwater, the brackish, and the marine environments. They have their preferences. Some algae, you will never see them in the marine. They are only in the freshwater. Some, they are in the brackish. Some, they are only in the marine. They don't survive in any other place. So they have their preferences, but these aquatic environments, they all have algae. Why? Because algae are the primary producers in all aquatic environments. Algae are the primary producers in all aquatic environments. So the primary producers must be there. If they are not there, nothing is going to happen in the water. No other life will survive in the water body when the primary producers are gone. So algae are very important. Now, apart from the aquatic environment, excuse me, apart from the aquatic environment, they grow in the snow. We have algae that survive in snow. We have algae that survive on bare rocks. We have algae in the desert soil where the temperature is extremely high. You know, this desert now, where there is no little or no water at all. We still have algae surviving in such environments, in hot springs. Imagine you remember Ikogosi, warm water spring. You'll be like, this water that is hot and the one that is cold, they meet together to form this warm spring. Will there be algae in the hot water, in quotes? Yes, we have algae surviving in hot spring. They are found living in lichen association. That word is lichen. If anybody should come and say leaching, we'll be in trouble. You don't go and disgrace us. It's lichen, L-I-C-H-E-N, lichen association, where you have algae in symbiotic relationship with fungi. So these are the habitat, the different habitats where you can find algae. Now, the economic importance, this we have talked about extensively in BTN 121. And I mentioned some of it here too. They are primary producers in all aquatic habitats. They form the base of the food chain by producing organic material from sunlight and carbon dioxide and water. This is basically photosynthesis. So, and that stems into the second point, the economic importance. They are responsible for oxygen production needed for aerobic respiration, because they release oxygen through the process of photosynthesis. You remember your photosynthetic equation. Oxygen is released as a byproduct. The oxygen released by algae in the aquatic environment help to sustain other organisms that need oxygen in the aquatic environment. So they are very important. They help to reduce global warming because they capture CO2. Don't forget your photosynthetic equation. Everything I've mentioned under economic importance is photosynthesis. CO2 plus water in the presence of sunlight, chlorophyll, all of these are present. Algae are able to capture the CO2. They are living in the water body, for example. They contain chlorophyll, that's why they can even photosynthesize. And sunlight is available. So photosynthesis is taking place. So they photosynthesize, they produce a carbohydrate and oxygen. So by capturing CO2, they are reducing global warming. Because CO2, carbon dioxide, is one of the major contributors to the global warming effect we have in the world now. If you have any complaint, please, again, type in the chat box. Some algae species serve as a source of nutrition. Some are edible. They have nutrients that are very, very, very useful for human health. They are highly nutritious. Not that you come to the lab and come and eat Spirogyra. For those that are edible, they have been experimented. Research have proved that they contain certain nutritional values that can help in improving human health. In countries like China, they feed on that. In Nigeria, I don't think anybody's, even me, I'm not eating algae. But then some are highly nutritious. Example is porphyra. Porphyra is a red alga. It's eaten as food in China and Japan. And like I said, it's not that they just went to the water, collect it and start eating it. Research has proved that those are edible and they are a source of nutrition. Algae has good source of arginic acid used in production of ice cream. Even if you have not eaten it before, like me, at least you have tasted ice cream. So the arginic acid used in the production of ice cream is can be sourced from algae. At least we are brushing our teeth now. Toothpaste, antibiotics, biodiesel, agar, they can be produced from algae as well. So if you have not eaten algae before, like me, and you hate ice cream, but you are using toothpaste. So one way or the other, you are benefiting from algae. And they cause algal bloom. I've said this before in year one. You should go and brush your notes again. This is just trying to do a kind of reminder for you. Algal bloom is when algae increase in large quantity because of excess nutrients in the water body. They now start reproducing in excess and they block the whole water body, discoloration of water, and this could lead to death of other organisms in the water body. So when you talk about economic importance, it's not just the good, though, it's the good, the bad, and the ugly. So those are some of the economic importance of algae. And like I said, these are not new to you. Algae and fungi similarities. This is important because you have been taught fungi. So what are the similarities they share? They are both thallophytes. That is, they both have undifferentiated thallus body. They are both seedless plants. They do not produce flowers. They share all of that. They exist. We have colonial algae, we have colonial fungi, we have unicellular algae, we have unicellular fungi. They both exist as filamentous and multicellular form. So they share that. They are both microscopic and macroscopic. Those are similarities. Differences. This is the very first one, is that algae are chlorophyllous, fungi are not. They don't have chlorophyll. Don't forget, these are everything I have said here is a potential exam question. As simple as they look to you now, I hope when you get to the exam, you will not go and be selecting the wrong answers. And also they are autotrophic while fungi are heterotrophic, of course. If fungi do not possess chlorophyll, at least they are going to feed somehow. So that is that. Classification. Now, this is not also new to us. I'm going to quickly brush through this. Classification based on different groups, classification based on pigmentation of photosynthetic pigments. The kind of photosynthetic pigments found in algae is one of the criteria used in classifying algae. Now, all algae possess chlorophyll A as their primary photosynthetic pigment. I said this in BTN 121. I'm repeating it again. All of them possess chlorophyll A, but then some have other accessory pigments like carotene, xanthophyll, phycobilins, in addition to chlorophyll A. Therefore, algae can be classified into different divisions based on that. All right. Moving on, we have the very first division Cyanophyta. They are commonly called the blue-green algae. You can also call them cyanobacteria. They possess chlorophyll A, which is green, and phycocyanin. The accessory pigment is phycocyanin. The primary photosynthetic pigment is chlorophyll A. The accessory pigment is phycocyanin, which is blue. That's where they derive the name from, blue-green algae. So you can call the group Blue division Cyanophyta, division Cyanobacteria, or you can call them the blue-green algae. You are still saying the same thing. And this is where you have your Oscillatoria, Anabina, Nostoc, Spirulina, and so on. Like I said, I'm giving you the very simple and very familiar examples so that you can remember. This is year 2. You don't want to stress anybody. So there are more than that. That is why you see and so on there. And so on is not an example for students now. They can and so on. And so on is telling you that there are more. We have division chlorophyta. They are commonly called the green algae. Please take note of the division, take note of the common name, take note of the photosynthetic pigments that are found in them, and the accessory pigments in the photosynthetic pigments. Remember, I already told you all of them have chlorophyll A, all of them, but they differ in their accessory pigments. For example, the cyanobacteria, the blue-green algae, they have phycocyanin. The green algae do not have phycocyanin. They don't produce phycocyanin. I hope that is understood. So the accessory photosynthetic pigment in the blue-green algae is phycocyanin, while the accessory pigment in the green algae is chlorophyll B. So that is the basis of differentiating them or categorizing them into different groups. You can't use chlorophyll A as a basis of classification. No, because they all have it. So how will you differentiate one from the other? But the differences in their accessory pigments help us to put them into different groups. So examples of your green algae are chlorophyll, chlamydomonas, volvox, spirogera. All these are common examples. I'm on the third one now, division Iglenophyta, your iglenoids. They are commonly called the iglenoids. The photosynthetic pigments are chlorophyll A, chlorophyll B, and carotene. You can see now, but there is already a difference. Chlorophytes or chlorophyta have chlorophyll B. They don't have carotene. But we have this in Iglenophyta.
Yes.
The rhodophytes are commonly called the red algae. Take note of examples, please. Everything on this note, on this lecture note, or in this lecture note rather, everything is a potential exam question. So please pay attention to the examples, the common name, the photosynthetic pigments. Please, very important. We have the division Rhodophyta. They have the chlorophyll A, chlorophyll D, carotene, phycoerythrin. Phycoerythrin is the red pigment. That is where they derive their name from, red algae. It's not that when you see them, they will be color red, though. They are not color red. Just because of this phycoerythrin is a red pigment color. It has it is pigmented red. So the rhodophytes are called red algae. Example are the Porphyra, the Batrachospamum. If you were in physical class now, you will pronounce that word, that name, Batrachospamum. Then we will see how people will be trying not to allow their teeth to fall out. But you still pronounce it when you come to the lab. Anyway, those are the rhodophytes. We have the brown algae division Phyllophyta. They have the chlorophyll A, C, carotene, xanthophyll. The xanthophylls here are lutein and fucoxanthin. Examples, laminaria, macrocystis, ectocarpus. There are other examples, but I've just tried to limit to 3 or 2 so that you are not bombarded. Then we have the diatoms. They have chlorophyll A, chlorophyll C, carotene, fucoxanthin. Examples, you have the navicula. I'm sure by now you are familiar with navicula, pinolaria, tabellaria, and so on. Those are the diatoms. This you will see in the lab. You will see the rhodophytes. No, we don't have specimen for rhodophytes. You will see the diatoms in the lab. You will see the green algae in the lab. You see the red algae in the lab. Those are microscopic. They are microscopic. They will prepare, slides will be prepared in the lab where you will see this ones. We don't have specimens for the red algae and the brown algae because they are macroscopic. They are macroscopic. We don't have specimens for them now, but you can see on my slides, on the PowerPoint I sent you, you will see what they look like. And you can also Google these names online. You will see what they look like. Okay, we have the dinoflagellates. That's division pyrophyta. They have chlorophyll A, chlorophyll C, carotene, and xanthophyll. You will say, but you said division pyrophyta have xanthophyll. Look at the type of xanthophyll in division pyrophyta is not the same as what we have in division pyrophyta. So those are the basis. That's the basis for basis for differentiation. Then we have the chrysophyta. That's the last one there, the golden algae. Don't think that they are golden color. These are also microscopic. Dinobrium is a very good example in this division. Classification based on food reserve. How do they store their food? How do they store their food? This is very important. It's very important. Chlorophyte food are stored as starch. The nature of primary product of photosynthesis is same in all classes of algae, but the nature of food reserve, how they store their food varies, differs. The food reserve are mostly polysaccharides. In chlorophytes, the food reserve is starch, like what we have in higher plants. Chlorophytes are very close to higher plants. They are microscopic, right? But they have some similarities with higher plants. A very good example is how they reserve their food. Cyanophytes and rhodophytes store their food as Cyanophysian and Floridian starch, respectively. Please, Daddy, don't make a mistake with that sentence. It means Cyanophytes store their food as Cyanophysian starch, while rhodophytes store their food as Floridian starch. That is why respectively is there. I hope you understand that, please, because these are potential exam questions. Don't lose marks unnecessarily. Don't come and say, excuse me, ma, it's just 2 marks for me to get to B. You earn every mark. So that's why I'm laying emphasis on anything that could be confusing. Okay. Please, can you state in the chat section if everything is okay so far before I continue? If it's okay, just give a thumbs up, or you say it's okay, just so I know that I am not talking to my, oh, God bless you. Thank you so much. Thank you so much. If you have any question, you can type in the chat section as well. Someone said, okay. Please, I sent the slides. Please ask your class rep. I sent my slides, the lecture notes, the PowerPoint. I sent everything through each class rep, please. Please contact your class rep for that, please. Thank you so much. Thank you so much. Okay, moving on to, okay, I've not finished that. Don't let me jump. In firefighter, the food is stored, the food stored is laminarine, the brown algae. The food, they store their food as laminarine and mannitol. You know why I said you should pay attention to examples given in previously? Because the question can go and take the name of a brown alga. It will just be like, example of a firefighter now. Just a microcystis stores its food as dash. The food reserve in microcystis is dash. If you don't know the division, that microcystis belong to, you are in trouble. You will be like, it's not in our notes, but it's there because I already stated that macrocystis is in the division Phytophyta. So I can decide to ask question, how it stores its food. That's why I said you should pay attention to examples. Everything in this note is important. This is not 100 level again, no. Because I know some people will be like, all these things is talking about. That's told us in year one now. It's wasting our time. Now we are testing your knowledge. We are going deeper. It's not just what is your name? My name is. It's no longer as straightforward as it used to be. Please pay attention. I can decide to say batrachospasmum, the food reserve in batrachospasmum is dash. If you don't know the division that batrachospasmum belongs to, you will be like, she did not teach us that one, no. But if you know that batrachospasmum is a red alga belonging to the division Rhodophyta, you will know that the answer to that question is Floridian starch. I hope you are getting it so that you don't just read and cram. Read with understanding, please, so that you don't lose marks unnecessarily. Okay. Diatoms store their food as leucocene. Imagine if a student does not know that pinolaria is a diatom. I now say, okay, question 2. Food reserve in pinolaria is dash. You now be like, pinolaria. From where? It's not in our notes. It's there, my brother, my sister, is there. Because I already told you that pinolaria is an example of a diatom. So you are able to say that, yes, food reserve in pinolaria will be leucocene. Please. Chrysophytes store their food as chrysolaminarine. Because you'll be expecting that I'll be like, rhodophytes store their food as that. That's too direct. That's 100 level kind of question. You are now 200 level students. So you have to show us that, yes, you have actually moved on to greater heights. Thank you very much for paying attention to that. In dinoflagellate, food is stored as starch, fat, and oil. The food reserve in euglenoids is paramillion. That is the second classification done and dusted, classification based on food reserve. Now we have a classification based on cell wall composition. What do they have? What do their cell wall contain? You see, they don't have the same components in their cell wall. Now we have the cell wall is usually made of cellulose. But the brown algae, for example, they have alginic acid. Remember when I said you can get alginic acid for the production of ice cream from algae? Is from the brown algae, it's from the cell wall of the brown algae that you could extract alginic acid. So the cell wall of brown algae, example, if we're in class now, can you type example of brown algae in the chat box, please? Let's know that we are together. Example of brown algae. Example of algae that will produce alginic acid in their cell wall. Example of brown algae. I can see people are scrolling down. Which one is brown algae again? It's just there. So I just need 1 or 2 names of algae that will have their cell wall containing alginic acid. Can you type, please? Is it that nobody knows the answer? Okay, thank you so much. Yes, fantastic. Somebody is typing xanthophyll. My brother, my sister, xanthophyll is not the name, or is not an example of algae. Xanthophyll is example of pigments found in algae, please. Thank you so much for your response. For your responses. I really appreciate that. Thank you, thank you, thank you. Please. No, somebody wrote kelp. That one is not correct, though. It's not correct. The giant kelp is the general name given to macroscopic algae, those algae that are not microscopic, those ones that are large. The kelp is the name, general name, giant kelp given to them. Kelp is not an example of algae. Please don't make mistakes here. Thank you very much. So it is very important for you to note that euglenoids do not have cell wall. Euglenoids lack cell wall. Your euglena, they don't have cell wall. Your phacus, PHACUS, lack cell wall. These diatoms have silicified cell wall. Their cell wall contains silica. The cyanophytes have mucopeptide cell wall. These are important for you to know. So the last one here is the nature of flagella. So we have motile cells. You know flagella is for movement, right? So you have motile cells of algae. They are called the flagellates. Now flagella types include thin cell and pantonymatic, thin cell or pantonymatic, and whiplash or acronematic. Now these two can be used interchangeably. It is important that you know that either you say the flagellum is thin cell type or pantonymatic. Or it is whiplash or acronematic. On my slides, you will see what they look like. The thin cell are hairy flagella, while whiplash are smooth flagella. You will see that on my slides. You can check that later on the PowerPoint slide sent to you. The type, quantity, and the point of insertion are main characteristics used in algae classification. When you have two similar flagella of equal length, they are called isochons. That is, if the flagella are both thin cell and they have equal length, for example, you call that isochons. If they are different with unequal length, you call that heterochons. Very simple and straightforward. You should also note that flagella are absent in red and blue-green algae. The red and blue-green algae, that's the rhodophyte and the cyanobacteria or the cyanophyta, do not possess flagella. Note that. Nothing should be ignored in this note. And that brings me to the end of the first lecture, and I move on quickly to the lecture 2. Can we go to lecture 2? Lecture 2 very quickly. That is our BTN 211 algae lecture 2. Now, if you are there, can you please type in the box section, open the second document. Type I am there so that I can move on. Please. Thank you, thank you, thank you. Thank you so much for your responses. Maybe we should be doing online class. Everybody is in their comfort zone, enjoying. Fine, thank you so much for your responses. Divisions and fighter. Here we'll be looking at selected divisions. We'll just try to go deeper. Not all of them, because when people are, hey, all of those divisions that she mentioned will not take them one. No. Just a few. This 200 level. Just a few that you will likely come across in the lab. and the few very, very common ones. So we just selected a few, and you can see that the notes are not plenty, no, just small, 200 levels, then they don't have time for stress. You don't stress them more. Yes, please. The first one, the division cyanophyta, they are prokaryotes. That's the only group of prokaryotic algae we have. Please take note of that. Please, please, please. I say that again, please take note of that. Like I said earlier, don't look at it like, hey, these things are very simple. People carry over this course and I don't know why. Because maybe they overlook every sentence is a potential question. So don't joke with it. The only group of prokaryotic algae are the blue-green algae. Others are eukaryotes. Why do we classify them as prokaryotes? They lack definition. You remember your bacteria in secondary school. You know, bacteria are prokaryotic. Why did we say they are prokaryotic then? The same reason applies here. And that is why the division cyanobacteria cyanophyta are sometimes called cyanobacteria, because they have the similarity with bacteria. They have this bacterial nature. The difference here is that they contain chlorophyll. They contain chlorophyll. They are chlorophyllos. And they manufacture their own food. But they still have these prokaryotic attributes or characteristics, like we say in biology. They lack a definite nucleus and membrane-bound new organelles. You remember your bacterial cells, you have all the organelles floating inside the cell. They don't have, even the chlorophyll in cyanobacteria or cyanophyta or blue-green algae, the chlorophyll is just floating inside the cell. The chlorophyll is not in a chloroplast. The chlorophyll is just floating in the cytoplasm. In other groups of algae, they have the chloroplast, and it is inside the chloroplast you have the chlorophyll. But in the cyanophyta, the chlorophyll is just floating in the cytoplasm, no chloroplast. So that is why they are prokaryotic. They are prokaryotic. They lack definite nucleus. They have membrane. They lack membrane-bound organelles. They resemble bacteria, hence they are sometimes referred to as cyanobacteria. Everything here I've explained. I've told you they are blue-green algae and why they are called blue-green algae. I've told you they lack chloroplast. But they have chlorophyll here. Bacteria don't have chlorophyll here. But then the chlorophyll here you have in cyanobacteria are not inside, the chlorophyll here is not inside chloroplast. It's just in the cytoplasm. This, I hope this is understood. They are unicellular, they can be unicellular, colonial, or filamentous, and examples are stated there. You can see is a very brief note, but like 4 questions or 5 questions or more can come from there. As I'm looking at this, how many lines? 1, 2, 3, 4, 5, 6. So don't joke with any information there. Don't go and be looking at the ceiling in the exam order. What is this one? What's the answer? No, 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 no. Just get it once and for all, and make sure you earn your A in the exam. It's very simple. I move on to division Hyglenophyta. They are unicellular flagellates found growing abundantly in polluted water. Where do you normally see Hyglenophytes? They are indicators of pollution. For those that have passed through Indomie Bridge. Some people, they don't know Indomie Bridge, and they are in Unilag. Hey, someone said I can't hear. Please, please go to where you can hear. Please. I hope, thank you for the person that responded that they can hear me. Please. I am very loud and shouting here so that you can hear me. I can only see 215 participants. People that are not coming for class. Hi. I don't want to say I reserve my comments, but then it is not a very good decision not to come to class. Okay, I said ignophytes they are indicators of pollution. Wherever you have polluted waters, be sure that you will see Iglina there. They enjoy, they flourish, they grow very well, they grow abundantly in polluted waters. And I was talking about Indomie Bridge. You don't know Indomie Bridge, next time you come to Unilag, ask. That place is very polluted. Anytime we collect water samples from there, we are sure that we will see Iglina looking very, very beautiful, green, and they will be running around because they are motile, because they flourish in polluted water. So they are unicellular flagellates. They lack cell wall, I've told you that, but they possess a structure called periplast or pellicle, which can be soft. So instead of their cell, a rigid cell wall, they have what we call periplast or pellicle. This is very important. This can be soft or it can be firm. You remember Iglina in secondary school biology, they have both plant and animal characteristics. This organism is both an autotroph and an heterotroph. We remember that in secondary, but why am I saying remember? Because some people might have forgotten, right? But please don't forget, it's still an extension of your biology, right? So don't forget. When they are acting as heterotrophs, Iglina surrounds the particle of food and consumes it by phagocytosis. You remember the way amoeba feeds? They just surround the food and absorb it into their body. So Iglina, even though they are not amoeba, they are plants, because algae are plants. I hope you know, if they are not plants, we will not be studying them in botany. So Iglina is able to feed through the process of phagocytosis, just engulfing the food through its cell membrane. I'm sure the feeding of amoeba, some people still do not know how it happens. It's on YouTube. There are so many information. All these things we crammed in secondary school, the videos are on YouTube. Go and search and see for yourself. Grow beyond cramming so that you will know that it's not a lie. Everything they were taught then, that we crammed and everything, you will see video, fantastic videos, not animations, real life videos of amoeba feeding through the process of phagocytosis. So please. Iglina has a long motile flagellum that uses to propel itself in its environment. So of course, you know that flagellum is for movement. So that is what it uses to move. When you see them, when you come to the lab, you will definitely see Iglina. Yeah, you will see them moving. That is if they are still alive under the microscope. You will see the way they will be moving around very fast. Is through the aid of their flagellum. So they have just one of that, not two here. This organism undergoes asexual reproduction by dividing longitudinally through the process of binary fission. So they just divide into two when they want to reproduce asexually. No form of sexual reproduction is known in Iglina. No form of sexual reproduction is known in Iglina. They are not known to reproduce sexually. Please, it is very important to note that. I move on to division chlorophyta now. They are commonly called what? Can you type in the chat box? The chlorophytes are commonly called what? and give one example. Don't look at your notes. Don't look at the notes. Type in the chat box. They are common, the chlorophytes are commonly called green algae. Thank you. Examples. I've not seen any example of green algae. Somebody said blue-green algae. No, I've missed it though. It's green algae. Yes, chlorella, thank you. Chlamydomonas, thank you. Spirogyra, fantastic. Volvox, great. Yes, thank you. Thank you, Volvox. Spirogyra, good. Spirulina is wrong. Spirulina is not a green alga, it's a blue-green alga. You see, please pay attention to this. Thank you very much for your responses. Thank you very much for your responses. Fantastic. Thank you. Please, for those that are still mixing it up, these notes are not, they are not voluminous, very short and brief. Please don't lose marks unnecessarily. Okay, I move on. Thank you for your responses. I move on to the division chlorophyta. Yes, they are found in freshwater ecosystem. Mostly, it's not as if you don't see them in the brackish or the marine, but mostly they are found in freshwater ecosystem. They may be unicellular, colonial, or filamentous that we have talked about. They can be motile or non-motile. So some possess flagella, some do not. Please, can you give me an example of motile green algae? You type motile, then you give me the example. Then you type non-motile, give me example. Please, can you make that very snappy? Example of motile green algae. Nobody's typing. Ah, God. It's very simple. Motile green algae. Examples. Yes, Chlamydomonas, of course. Thank you. Volvox, good, great. Thank you. Good, fantastic. What about non-motile? What about non-motile? What about non-motile? Can I have non-motile? Pandorina, Eudorina, the examples of motile. Yes, Spirogyra, no flagellum. Oh, yes. Then chlorella is non-motile as well. Chlorella, I was expecting, yes, great, thank you. Somebody just typed chlorella non-motile. Fantastic, thank you so much. Thank you for your responses. Okay, that's fine. So, they possess definite plastids called chloroplasts. Now, their chloroplasts have various shapes. Yes, the chloroplast can be cup-shaped, spiral-shaped, oval, or plate-like. Can you give me an example of spiral? Oh God, I just mentioned it. Example of alga with spiral-shaped chloroplast. I almost mentioned it. That's Spirogyra. You know when you draw your Spirogyra and you put those intertwined threads inside that you draw like 8, 8, 8, 8. Those are the spiral-shaped chloroplast. You have cup-shaped chloroplast in chlorella and Chlamydomonas. Yes. Yeah, this is just 200 levels, so you don't, we are not going too in-depth, but that is something you should know for the purpose of the exam. So some people have been drawing Spirogyra in their dreams and they don't know that that spiral stuff you put in the middle is the chloroplast. They will label it though, but they never paid attention to the fact that it is the spiral-shaped chloroplast. So it's a spiral-shaped chloroplast. So we have different chloroplast shape in green algae. Asexual reproduction is usually by formation of zoospores, which may be formed in vegetative cells, while zoospore formation is completely absent in the other conjugalis. So examples are there, you're already familiar with that. So we look at the very good example here, Chlamydomonas. Why did I choose Chlamydomonas? Because we have been hearing it from our secondary school biology. So let's look at how it reproduces, asexual reproduction. is by zoospore formation under favorable condition. When the condition is favorable, they reproduce asexually. Yeah. The parent cell comes to rest and the flagella are cast off. They will lose their flagella. When it is time, when they decide we want to reproduce asexually, chlamydomonas, they will come to rest. All the moving around will stop. All that active moving around will stop. They will lose their flagella. When they have lost the flagella, what will they use to move or hold? So then the contractile vacuole disappears while the protoplast is withdrawn from the cell wall. The parent chlamydomonas cell undergoes a longitudinal division, giving 2 daughter protoplast cells, while the chloroplast and the prinotes also divide. This is on the PowerPoint, is in the PowerPoint slide. The life cycle is there. If you look at it, at every stage is represented in the note. So you don't have to cram the life cycle, just understand the note and know where what is happening at each stage. Each daughter cell subsequently undergoes another longitudinal division. You have the series of longitudinal division to give rise to 4. So you have the parent cell divided into 2 daughter cells. Then you have the 2 daughter cells dividing into 4. So each one will divide into 2 again. So you have 4. All these divisions are mitotic. Thereafter, the daughter protoplast form their own cell wall. They now produce their own flagella, develop into zoospores. The zoospores are liberated in the water by the rupture of the parent cell wall or by the gelatinization of the cell wall. So all of this is happening in the parent cell wall. All the divisions are happening in the parent cell wall. By the time the process is completed, the cell wall will rupture and the zoospores are what? They are released. So from 1, you have 4 cells coming and they will germinate into adult chlamydomonas. The life cycle illustrating that is in the PowerPoint. It is not difficult, but sometimes when people hear life cycle, they're God, it's very simple. I've simplified it. It goes line by line with the note. Sexual reproduction occurs when the conditions are unfavorable, like when the weather is too hot, or the nutrient is low, or any condition that is not comfortable for chlamydomonas will result into sexual reproduction. They will not carry out asexual reproduction when the conditions are unfavorable. You should take note of that. If you look at the note very well, you will see that I highlighted that. I highlighted that so that you will know that it is important. So that when I ask you, during unfavorable conditions, how does chlamydomonas reproduce? You now be like, is it sexual? Is it asexual? I know it's between C and D. I don't know whether it's C or D. Then you start doing 1, 2, 3, 4. God, where I stop 4, it should be the answer. It's unnecessary. Even God will be wondering, what is this my son or daughter doing? So just know it now that when the condition is unfavorable, reproduction in chlamydomonas is sexual. And the process is stated there in the note. The adult cell, again, the flagella is withdrawn. You should know that Iglina have 1 flagellum, chlamydomonas has 2. I mean, I just said to state that now. Okay. Before reproduction, they withdraw their flagella, the protoplast divide, the same thing. So several times to form 8 to 32 biflagellated gametes. These gametes are without a cell wall and look exactly like zoospores, except for their smaller size. So the gametes here, they are smaller than zoospores. You know, zoospores is what is formed during asexual reproduction in Chlamydomonas, but here, gametes are formed and they are smaller in size than the zoospores. The gametes are alike in size, structure, and physiological activity. They behave the same way, so they are called isogametes. After liberation, gametes derived from different individual cells usually fuse at their anterior end to form quadriflagellated zygote, which undergoes meiosis to produce zoospores, which germinate to give rise to mature Chlamydomonas. The life cycle showing the sexual reproduction is also on my slide. Please, this note on sexual reproduction follows the life cycle step by step. No need to cram anything. It's just for you to understand it. So when they join at the anterior end, one is acting as male, the other is acting as female. That is why it's sexual reproduction. I hope that is clear. I move on to division bacillariophyta. Bacillariophyta, the diatoms. Examples of diatoms apart from navicular. Nobody should type navicular. Nobody should type navicular. Examples of diatoms apart from navicular. Nobody is typing, no. Nobody is typing, no. Somebody is laughing. What's funny? Thank you, Pinularia. Thank you for spelling it correctly. Pinularia. I said nobody should type navicular. Hey, Tabellaria, thank you so much. That's fantastic. Thank you. Cimbella, this is a phycologist. So how did you know Cimbella? Great. Thank you so much. Thank you so much for your responses. Thank you so much for your responses. Now let's go back to the note. Division bacillariophyta. They are common in the marine and freshwater environment. Take note, I didn't say that is where they are found, though, only. Common. That's where you usually see them. So in your gutters, in your drainages, they are everywhere like that. So you have the diatoms, even in the soil. We have diatoms, we have algae in the soil. I've mentioned that earlier. So when you come to the lab, you will definitely see diatoms. Definitely. Yeah. Diatoms serve as food for several aquatic animals. They are very fascinating when viewed under a microscope. Now I'm going to take you quickly to the PowerPoint. Can we go to the PowerPoint where we have the diatoms so that we can look at it? Scroll down to diatom cell. Yes, scroll down to diatom cell in your PowerPoint slide, please. I don't know why my screen is not sharing. Please, if you are there, please, can you type I am there? You go to PowerPoint. Thank you so much. The PowerPoint where we have the diatom cell. The diatom cell. Good. Can you see the epitheca, the hypotheca, the raph? Those are the image you have where you have scanning electron microscope image. That is not what you will not see diatom like that in the lab. No, this one is from a more specialized microscope, not the kind of the microscope we have in the lab here will not show you this kind of image. The microscope we have in the lab here will show you the image on the previous slide where I have examples of diatom. You will see your navicular like that, your Pandaria like that. But I want you to pay attention to the diatom cell now. Diatom cell. Now, you can see the epitheca, the hypotheca, the raph, the fusto. So the whole diatom cell is like, I hope you remember your St. Louis sugar box. Hey, that's if they know what St. Louis is, so Gen Z. Gen Z, do you know St. Louis sugar, the box? Please, if you know St. Louis sugar box, please type in the chat box, please, so that I will know. Hey, somebody say I don't know it. When you get home, ask your mommy. Gen Z, they
Check
don't know
Google.
sugar box. There is this brand of yes, blue cube with lion image. Thank you so much. Thank you for that description. That one is a very good Gen Z. Now, rich man sugar. Are you a Gen Z? This person that said now rich man sugar. How did you know? I don't expect Gen Z to know St. Louis. Okay, fine. Thank you. Do you know the way the box is? That is what I will use to describe the diatom cell. You see the way the upper part of the box fits directly on the lower box. So you see the lower box is a little smaller than the upper part of the box. That's the same way the diatom cell looks like. Although it's not a rectangular box now. I'm just trying to tell you that the upper layer fits directly on the lower layer, just as that St. Louis sugar upper part fits on the bottom part of the lower level box of the St. Louis sugar box. So the lower layer is called the hypotheca. Very important, please. This part, question it will flow, like water. The lower part of the diatom cell is called the hypotheca, while the upper part is called the epitheca. Now, the raft there is what they use for movement. Diatom cells move. They don't have flagella like as discussed in Chlamydomonas and all of those. But diatom cells, they move with the aid of raft. Now, the whole diatom cell, the whole cell, like you have the whole St. Louis sugar box, the whole cell is called fructose. The whole cell is called fructose. That is describing the morphology of the diatom cell. Yeah, that is very, very important. Please go to the next slide, that is the fructose of the diatom. The fructose of the diatom. You can see that is like they opened the cell, the fructose, into two, showing you the epitheca, the hypotheca, and the band, the Gadu band in between them. Yes. So that is a pictorial representation of what the diatom cell looks like. So when I ask you in the exam that the whole diatom cell is called dash, you know what to say. Can we go back to our lecture note 2? So that I can continue, and are we there? Please, if you are there, kindly type I am there. Let's go back to our lecture note 2. Thank you so much. Thank you. Thank you so much for your responses. Thank you so much for your responses. Okay, moving on. So I said the diatom cell has 2 valves. The whole cell is the fructose, the upper layer is the epithecal. It fits closely over the bottom half called the hypothecal. And the diatom cell has 2 valves or halves made up of silica. Diatoms have a structure called raft. It's not Raphael, it's not Raphael, it's raft that allows them to move over surfaces. So the raft aids in movement. So we have 2 groups of 2 subclasses of diatoms. We have 2 subclasses. They are usually grouped into 2 subclasses. We have the centrioles and the pennials, based on the symmetry of the fructose. Now, the centrioles, they are radially symmetrical. That is, the diatom cell fruit stool can be divided into equal halves by cutting through any plane. Whether you cut it horizontally or vertically, you will get two equal halves. Any diatom that can be divided into two equal halves, whether cutting horizontally or vertically, that belongs to the subclass Centralis. Those that are bilaterally symmetrical, they can only be cut through one plane. It is either you cut them horizontally or vertically to get two equal halves. Take note of the highlighted words there. Equal halves, getting two equal halves is the basis and the plane. As human beings now, are we bilaterally symmetrical or radially symmetrical? If we were diatoms, if you understand what I just said, if we were diatoms, which group will you put us? Tenalis or Centralis?
Tenalis.
Can I have responses? Good, fantastic. Bilaterally symmetrical, because we can only be cut in one plane to get two equal halves. If you cut human being from the belly button, what you have up will not be the same as what you have below. But if you cut human being into two, like from head down, you will have two equal halves. So that can help you to remember what's the difference between the two subclasses of diatoms. If you don't get it, read it again. It's very simple and straightforward. So our reproduction in diatoms can be sexual or asexual. Reproduction in diatoms can be sexual or asexual. I'm going to be a little bit very fast here. So asexual reproduction in diatoms is through the process of cell division. That is, the cells will divide. During cell division, two daughter cells are formed in which one is smaller than the other. The nucleus divides first, followed by chromatophores and prenoids. This is followed by longitudinal division of the protoplast parallel to the valves. The two daughter protoplasts separate so that one of them gets the epitheca while the other gets the hypotheca. So you have a fruit stool of diatom, which is the adult diatom now. You know the fruit stool has the epitheca and it has the hypotheca. When that adult diatom wants to undergo asexual reproduction, the fruit stool will open, will separate into two, epitheca, hypotheca. So as that separation takes place, you already have two daughter cells. The two daughter protoplasts separate so that one gets the epitheca. When the diatom fruit stool opens, you have epitheca and hypotheca, right? So one daughter cell will be the epithecal, the other daughter cell will be the hypotheca. In both cases, the parent valves function as the epithecal of the daughter cell. That the protoplast will now develop hypotheca. That hypotheca of the daughter cell that got the hypothecal valve of the adult will become the epithecal of the daughter cell. It will develop hypotheca. That was why I said in both cases, the parent valve functions as the epitheca of the daughter cell. And the new valve formed is always the hypotheca. Both will still develop another valve. That's the other half. The other half that they will develop as daughter cells will be the hypotheca. While the half they inherited from their parents or the adult cell will function as the hypotheca. Will function as the epitheca rather. If this is not clear, when we come for physical lab, I will say that again and give you the explanation more clearly in the lab. So that is that concerning that, but it is pretty straightforward. Continuation of this process results in progressive reduction in size, since the hypotheca, which is smaller, of the parent becomes the epithelial of the daughter cell. So it's going to get to a point that the diatom cell becomes too small. It will not be able to carry out asexual reproduction again. It won't be able to divide again because the cell becomes smaller and smaller. Because the hypotheca of the adult is always becoming which is smaller, you know, the lower valve is smaller than the upper valve. So it is already small. So continuous division will lead to decrease in size, continuous progressive reduction in size. So when the datum cell cannot reproduce by cell division again, when it is too small to divide, it will result to sexual reproduction, which leads to formation of oospores. So sexual reproduction in datums is by formation of oospores. Yes. So economic importance of datums are stated there. They are very good when it comes to assessment of water quality. Example, navicular has been used extensively for the assessment of water quality. Diatomaceous earth, I need you to go and read about that. Is a huge deposit of datum siliceous cell wall at the bottom of the water bodies. So diatomaceous earth is commercially useful in sugar refineries. So when the atoms die, they fall to the bottom, they sink rather, I think that's a better word to use, they sink to the bottom of the ocean. So after a period of time, you have their deposits there, it just keeps on piling up. This is called diatomaceous earth. It is useful in sugar refineries for the filtration of liquids. It is used as an inert material filter in production, in products like plastic. Furthermore, addition of diatomaceous earth to paint increases their visibility at night. The paint that we use painting our buildings. They are also used in the manufacturing of toothpaste and silver polishes. So those are some economic importance of diatoms. I move on quickly to division phyophyta. Division phyophyta, these are not microscopic. They are not microscopic, they are macro. These are the giant kelps. They are multicellular macroscopic forms. They are mostly found in the marine ecosystem. You hardly find them in fresh water. They are the seaweeds. They are the sea, that's their habitat. Brown algae are distinguished from other algae by their characteristic brown coloration due to the presence of pigment fucoxanthin. If you go to the PowerPoint slide, you will see a man holding a brown algae. You see that, laminaria. That is still a thallus, so regardless. You see them, they are washed to the shore. So that's an example of laminaria, a brown alga. I go back to my note, which occurs in association with chlorophyll. I've stated that earlier. Many species are attached to rocky surfaces, while some are found at the seabed, and some grow as endophytes or in epiphytes in other algae. There are no unicellular or filamentous forms. All of them are multicellular. I already said that. The simplest brown alga is ectocarpus, while some members of this division have a talus which is nearly 100 meters long, regardless of how long or how large they are still talophytes. I think I've said that like how many times today? Okay, divisions Phyllophyta contain some of the largest algae in size. However, they lack vascular tissues. You know, your higher plants have vascular tissues. Reproduction takes place by vegetative, asexual, or sexual method, which are stated there. So, those are self-explanatory for the type of reproduction. You should take note of the one that is looking very plenty here. Asexual. That's most likely where the question will come from. Don't quote me. I said most likely. Okay, you can read that up, but then know that reproduction can be vegetative. They can break into two by fragmentation. Yeah. And each individual, the adult will break into two. Each individual will now develop into adult microalgae. That is possible. Then asexual, through zoospores, and the sexual by formation of gametes. I move on to division rhodophyta. Division rhodophyta are exclusively marine. They are also macroalgae, macro, not micro. Only a few are found in the fresh water. Most of them, they are marine. Most of them are multicellular. They are attached to the bottom of the sea. They are distinguished from other algae by, take note of this. How do they differ from other algae? First one is the presence of red pigments called phycoerythrin. I've said that before, which are the accessory pigments in addition to chlorophyll A and B. They do not have motile cells. Motile cells are completely absent in this division. So there is no cell with flagella, flagellum. No. There are no motile cells in this group, and they have highly specialized sexual reproduction method. Yes, those three are very important because they distinguish rhodophyta or rhodophytes or red algae from other algae. Examples, we have Polysiphonia, we have Batrachospermum, we have Porphyra. And another thing you should take note of is in Polysiphonia, there are 3 distinct stages represented in their life cycle. Now, this has been summarized. The people that will do, the 300-level student that will come and do phycology, they are the ones that will learn this in details. But for your level, just know the 3 distinct stages in the life cycle of red algae Polysiphonia. And with that, I have come to the end of my lecture today. Yes, thank you very much for posting the slides. So, any question? If you have any question related to what I have taught, you can type in the chat box. Meanwhile, while I'm waiting for, don't ask me questions about student funders. I just said related to what I have taught. Because I don't want anybody to come and ask me when is student fund or something. I don't know what they even call it. I be loan. I don't know anything about that. Please. So while I'm waiting for questions, if there are any related to what I have taught today, I will want to let you know that if you have not purchased your lab manual, you are on a long thing. Oh. This is where trouble used to start. I have sent that information across like 2 weeks now, if I'm not mistaken. I am going to send the assignment through your class reps today. Before the end of today, you will receive message from your class rep on what assignment you are going to do in your lab manual. And the mode of submission will be stated, will be communicated to you through your class reps. The date of submission as well, which is this December. The mode of submission and the date of submission is this December, which could be before the end of this week. So please, if you have not purchased your lab manual, you had more than enough time to do that. Please purchase your lab manual ASAP. Please. I'm saying please now so that somebody will not come and be saying please. Can you please explain the assessment of water quality using navicular? So navicular does not have that letter R. It is spelled without the letter R. So, like I said, algae and plants generally, they are some bioindicators. They are used for biomonitoring. Like when I told you, thank you for correcting that. When I told you that euglena is an indicator of pollution. If you, for example, go and collect water sample from Indomie Bridge now, and I did not know Indomie Bridge before. Maybe I'm in America and you collected water sample from Indomie Bridge. I don't know Indomie Bridge. I don't know whether it is polluted or not. You just collected water sample and you send it to me that I should help you to check what is happening there. Immediately I prepare a slide from that water sample and I see euglena in abundance. The next thing I will say is to ask you, is that water body polluted? And your answer must be yes, Because with that population of euglena, there is no way the water body is not polluted, because euglena is an indicator of pollution. They are known to grow very well in polluted water bodies. So for navicular, you are going to check Google, just as I've used euglena to explain to you. So what does navicular, if I see abundant navicular in my water, what will it tell me? So I'm going to direct you to check that up. Is it that when navicular is plenty, does it mean that the environment is polluted or the water body is safe for drinking? That is biomonitoring, that is water quality assessment. Another example I will give you, but not the navicular, is batrachospasmum. Batrachospasmum is a red alga. If you see batrachospasmum in a water body, that water body is safe for drinking. You don't need any other, analysis. Because if batrachospasmum can survive in that water, growing abundantly, that water is safe. It's not just clean, it's safe for drinking. Because batrachospasmum can only survive in water bodies that are free from pollution, not the one that you go and urinate inside or poo-poo inside. If batrachospasmum was growing there before, immediately that pollution comes in, they will die. That's another form of assessment of water quality using algae. I've given you two types of algae now. The one that tells you that that place is polluted without you going there. Tell me it is polluted without telling me. When I see Iglina in abundance, that water is polluted. There are no two ways about it. If I see batrachospasmum in abundance, then that water body, I can drink it without thinking twice, because batrachospasmum cannot tolerate any kind of pollution at all. So you tell me, because now we are in university, we have to be led. So what does navicular indicate? Is it that the water is polluted or not? Thank God I know your name. I will go and come and look for you. I do praise. I hope I have answered your question. I know you will say no because I didn't tell you navicular. That's why you're in the university. But you already know what water quality assessment is all about. So with that knowledge, as an undergraduate student of the University of Lagos, I'll be waiting for your response. So what does navicular indicate? It's a bioindicator. It can be used to assess the quality of the water. Is the water safe for drinking or is it not safe for drinking? Is it safe for somebody? Is always I do praise that is so still laughing. You did not even tell me whether I answered your question or not. Any other question? Please go and get your lab manual assignment. I'm sending it today. And the mode of submission will be communicated with the message. Date of submission will be communicated with the message. So, any other question? Please, you can unmute yourself, and because the class is almost over, please, Ma, can you explain the reproduction in that? Or I have done that. If you did not understand that, you can ask me when you come for physical lab or physical. No, I'm not taking physical class again because I am done teaching. But when you come to the lab, I will definitely be there by God's grace. So you can ask me your questions there. What's the meaning of plurilocular sporangia? Fantastic. So you have unilocular. Uni is one. Pluri is like plural. If you look at the notes very well, you will see the difference between the two. Like I said, there are some things will not bother you about because this is just 200 level. So those ones produce two types of sporangia, plurilocular sporangia. You remember sporangium? Sporangium is singular sporangia. It's just that where the spores are produced. Aha. So please, can you unmute yourself? At least the class reps, so that I will know whether I can leave the class now. Any question? Is it that people cannot unmute themselves? I can't hear anybody talking.
No question, ma. No question.
Hey, okay, now I can hear you. You said what?
No question.
No question, ma.
No question, ma.
Okay. Thank you very much. What of those that live far, that can't be manual today? I have told you to buy manual for how long? You are telling me. I've told you to buy manual since when? Who is this person? I hope he's not here. So. He said you can't buy manual today. Do you know since when I've sent the message? My sister, Mark, some of us don't stay in Lagos. Send your friend to buy the manual for you. Maybe you don't have friend that stay in Lagos. Go and get your manual. Get your manual. You have friend. Those that bought the manual, I hope the manuals are still available. If in case manual is out of stock in the bookshop, please, class rep should let me know so that I can tell my HOD. If you do not stay in Lagos, send your friend to buy your manual for you.
Okay, I think
Yes, lab will be physical. I've not said anything about lab today. I said assignment will be given to you from the lab manual, and it is very important that you get it. Physical lab is going to happen in January 2025 by God's grace. But prior to that, you have to get your lab manual because I'm sending your assignment today before the close of today. And mode of submission and date of submission will be communicated through your class reps as well. So thank you so much. For further questions, when you resume physical classes, I will be in the lab by God's grace. You can ask me your questions. So enjoy the rest of your day and thank you for coming. Bye-bye. Somebody said I'm in the US. Okay.
That one is actually joking. Ma, I want to ask a question. Wow. Hey. Come on. Oh, gosh.